Namaste students, I am Jeevan Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic Class 2 MHC molecule. In the previous video, we discussed about the MHC molecule, what is the MHC, major histochemical complex, and the structure and function of the Class 1 MHC molecule. So Class 2 MHC molecule is present on the surface of antigen presenting cell and cell which engulfed the foreign antigen. It is present on the surface of the antigen presenting cell and the cells which have engulfed the foreign antigen. It binds with the exogenous antigen, the antigen which has been degraded. Endocytic antigen means this antigen which have been degraded by the internal parts of the cell, and it binds histocompatibility complex number 2 MHC2 bind to the CD4 molecule, and CD4 molecule is present on the surface of the T helper cell. CD4 is a receptor of the T helper cell, so by a domain the T helper cell attach to the MHC class 2 molecule with the help of the CD4 receptor. It also consists of two polypeptide chains. Here, two polypeptide chains are there, alpha chain and the beta chain, and four domains: alpha 1, alpha 2, beta 1, beta 2. Antigen is processed inside the endosome, and peptide is further loaded on the groove of the MHC class 2 molecule. So this is the MHC class 2 molecule, and that's an introduction of MHC class 2 molecule. Now going to structure of MHC molecule. What is the structure of MHC class 2 molecule? The class 2 MHC molecule consists of two polypeptide chains, namely alpha chain and the beta chain. Alpha chain is 33 kilodalton and the beta chain is 28 kilodalton chain. The both chains are attached non-covalently to each other. Each chain contains two units. The two units of alpha chain are called the alpha 1 and alpha 2. The two domain of beta chain are known as the beta 1 and beta 2. The beta 1 and alpha 2 are transmembrane domain, which anchor the MHC to plasma membrane. The alpha 1 and alpha 2 domains jointly create a peptide binding group. So in the upside, there are alpha 1 and beta 1, and in the bottom side there are alpha 2 and beta 2 domains. So I must share the screen so that the students can know the structure of the MHC class 1 molecule, class 2 molecule. It is MHC class 2 molecule, class 2 MHC molecule. It has alpha 1 and alpha 2. Four domains are there, alpha 1 and alpha 2. Alpha 1 is here in the single chain, and beta is a next chain, two chains, beta chain and alpha chain, and four domains, alpha 1, alpha 2, beta 1 and beta 2. The alpha 1 and alpha 2 hold a antigen, and beta 2 and alpha 2 are the transmembrane domain here. They have a proteins with transmembrane domain. This is the disulfide bond, and they are bound to each other by the non-covalent bonding. This is the membrane proximal region, it is membrane distal region, it is transmembrane region, and it is cytoplasmic tail. This is the region where CD4 cell will bind. here. CD4 cell will bind here, and this is the extracellular space. The peptide will bind here. Okay, it will flank here. It is not anchored in the MHC class 2 molecule. The peptide will flank here. Means only one single end will bind to the binding groove. This is the binding groove, and only one end of the antigen will bind here. So this is the structure of MHC class 2 molecule. Now some idea related to the class 3 MHC molecule. The molecule include the complement like C2 and C4, and vector B also are included in the class 3 MHC molecule, which take part in the complement 6 fixation. And the class 4 MHC molecule, these molecule is present on the T cell of leukemia as well as the immature thymocytes. So, but they are not used usually, and little knowledge is available about the class 3 and the class 4 MHC molecule. Now function. Talk about the function of MHC class 2 molecule and MHC molecules. MHC molecules are loaded on the sample peptide fragment derived from the degradation. First, an antigen will come, and we have learned about the phagocytosis process. When an antigen encounters into the body, antigen presenting cell will engulf the antigen, and it will make some peptides of those antigens, and major histocompatibility complex will present these uh, peptides to the uh, T helper cell and the cytotoxic cells. MHC molecule contains self as well as non-self antigens. They bring about defense against the infection and the diseases, and they mediate certain autoimmune disease also if they recognize self particles as a non-self. And they are responsible for the individual smile of people. If I have something some smile, then it indicates that this smile is due to MHC molecule. If somebody has any kind of smile, this smile will be only due to the MHC molecule of particular human. So this is all about the class 2 MHC molecule. Now we need to discuss about the processing of endogenous antigen by the MHC molecule. So for that, I need to share the screen here. This is the endogenous antigen. First, what happens? Endogenous antigen means the antigen is inside our body. First, this is endogenous antigen come to the body, and one proteasome. This proteasome will degrade this endosome. This proteasome will degrade this endogenous antigen into small particles, and after that, transporter associated protein will move these peptides to the endoplasmic reticulum. So this is the endoplasmic reticulum, and on this endoplasmic reticulum there are some MHC molecule, class 1 MHC molecule. Each class 1 MHC molecule will be there on the surface of the ER, and there is a major binding groove, peptide binding groove on the surface. So on the peptide binding groove, These MHC class 1 molecule will bind a peptide and then they will move to the Golgi body. This is the Golgi body. After binding to Golgi body, this Golgi body will make some complex structure of this MHC molecule and make a phagosome here. This phagosome is created after Golgi apparatus. Then this MHC molecule will then be transferred outside the cell, and this MHC molecule now will present this antigen to the another cells, that is cytotoxic cells on the surface of this cell, this antigen presenting cell. So first, and the endogenous antigen will come and make some uh, go to the proteasome complex. There will be some formation of some peptides, and these peptides will then move to the ER where MHC molecules are there, and then MHC molecule will present these peptides to cell, another cells outside the body, outside the cell. So this is the end processing of the endogenous antigen. Now processing of the exogenous antigen and first an exogenous antigen will come and it will be phagocytosed. It will be phagocytosed by phagocytic cell or antigen presenting cell. Then this antigen will be digested by some lysosome, and a phagolysosome complex will be formed. After that, this antigen will be transferred to the MHC endoplasmic reticulum where MHC molecule will be there. Here, ER molecule will transfer this protein and peptide to the MHC molecule, to the Golgi complex. This Golgi complex will finalize the peptide and it will make some more digestions. It will make some complex structure of peptide. This after complex formation of this peptide, this MHC molecule will make some phagosome complex, and this phagosome complex will be then transferred outside the cell and will be present it this antigen into the another cells which are MHC class 2 molecule to the T helper cells. So this MHC class 2 molecule will show the antigen to the T helper cell. This can also be done in another way also. First, this exogenous antigen will come and it will be digested by the lytic enzymes, and that after that a lysosome complex will be formed. After that, this MHC molecule will come and it will bind the antigen on the peptide binding group, and after that this peptide along with the MHC molecule will be moved outside the cell, and this time the MHC molecule will show this antigen to T helper cells. So this is all about the MHC molecule. So students will be able to understand what is the MHC class 2 molecule and the process of antigen presentation by the MHC class molecule. So my lecture is over here. What kind of questions can be asked here? Explain major histocomparative complex class 2 structure and function. Explain the processing of endogenous antigen. Explain the processing of the exogenous antigen. So this is all about the MHC molecule. So my lecture is over here. Thank you very much.